Alzheimer's Drug Discovery Foundation hosted its 9th International Conference on Alzheimer's Disease Drug Discovery on October 6th to 7th this year at the Westin Hotel in Times Square in New York City. The conference promoted networking to catalyze the exchange of ideas and to foster alliances to accelerate the development of new treatments for Alzheimer's disease and related dementias. This year's meeting attracted a multidisciplinary network of over 170 academic, industry, and government researchers. While the conference centered on many different drug discovery efforts led by top investigators in the field of neurodegenerative diseases, neuroprotection and synaptic enhancement led the way as a means to prevent and potentially reverse the cognitive decline associated with Alzheimer's disease. Both days of the conference began with plenary talks by Dr. Ottavio Roncio from Columbia University and Dr. Thomas Lance from Pfizer on new molecular targets in Alzheimer's disease to enhance signaling pathways involved in learning and memory. The first session of the conference focused on neuroprotection and included talks on targeting neurotrophin receptors, cell signaling pathways involved in synaptic and dendritic maintenance, learning and memory-induced gene expression, and novel treatments that may rescue cognitive impairment by inducing neurogenesis. The key pathological hallmarks of Alzheimer's disease, amyloid plaques and neurofibrillary tangles, continue to be a major focus of this conference. The second session concentrated on targeting beta-amyloid aggregation and protein misfolding and included presentations on targeting PPAR gamma, ApoE, chaperone proteins, and using curcumin-like compounds to promote clearance of beta-amyloid in Alzheimer's disease. The third session focused on neurofibrillary tangle-targeted therapies and consisted of presentations on disrupting tau aggregation and or phosphorylation using small molecule inhibitors, enhancing degradation of tau aggregates, and using tau-focused vaccine therapy. Finally, anti-inflammatory and antioxidant strategies were outlined in the last session. Inhibitors of oxygen-generating enzymes and of protein kinases and cytokines involved in inflammatory responses were discussed as a means to prevent neuronal damage. The conference concluded with a talk by Dr. Musa Houdim on designing drugs that hit multiple disease-relevant targets. Throughout the conference, attendees also learned of exciting new treatments that are progressing into human trials. Dr. Juan Sanchez-Ramos is testing a growth factor that promotes neurogenesis and microglial activation in a small human trial to ascertain safety and efficacy. In addition, Dr. Randall Bateman spoke about methods for monitoring beta-amyloid in cerebral spinal fluid of human subjects, which could be further developed to monitor efficacy of new treatments. Lastly, Dr. Ileana Gozes from Milan Pharmaceuticals spoke about her company's nasally administered neuroprotective peptide that is now advancing into phase 2B trials for Alzheimer's disease. Overall, the extensive array of drug discovery research presented at this conference will contribute to novel directions for Alzheimer's disease drug discovery and may ultimately lead to new therapeutic treatments for the disease. We hope you will join us in 2009 at the Drug Discovery for Neurodegeneration Conference in Washington, D.C. In addition, stay tuned for more information on the 10th International Conference on Alzheimer's Disease Drug Discovery. For more information, you can visit www.alzdiscovery.org.